Our last talk of the afternoon is from Andrea Brand. Are we cured? Yeah? Okay. And she's going to talk about challenges and lifestyle in the lab.
She, as many of you know, became the head of the Red Cross. Many of us think she did a fabulous job. They booted her out. She was very vocal, and when they fired her in the press, she said, "They think they hired Mary Poppins, but they got Jack Welch."
Should we give it a go? Well, thank you for the invitation to come and speak. I have to say I was not quite sure how to balance the talk, so forgive me if it's a little too much of one thing or too much of another. So my title is Work-Life Balance, Studying the Brain Without Losing Your Head. And I have to say, when I told my lab that I was going to talk about work-life balance, they all fell over laughing, so that gives you an idea here. So I'm going to try to give you a little bit about life and a little bit about work. So what we're interested in is the brain, the nervous system. And so I guess the first question is, why study the brain? And I'm going to show you a couple of quotes that I rather like from Hippocrates. So men ought to, and I should say this does say men, we can change it to men and women, ought to know that from nothing else but thence, from the brain, come joys, delights, laughter, and sports, and sorrows, griefs, despondency, and lamentations. And by this we acquire wisdom and knowledge, and see and hear, know what are foul and what are fair, what are bad and what are good, what are sweet and what unsavory. But he went on to say that by the same organ we become mad and delirious, and fears and terrors assail us, and ultimate and untimely wanderings, and ignorance of present circumstances, and unskillfulness. And all these things we endure from the brain when it is not healthy. In these ways, I'm of the opinion that the brain exercises the greatest power in the man and the woman. And I particularly like this one because when it comes to work-life balance, this ignorance of present circumstances reminded me of when I walk out of my office to do something and immediately forget what it was I was going to do. And I gather I'm not the only one this happens to. So just to give you a little background about what inspired me to go into science. So firstly, I was born in New York, but my parents are English, and I have a slightly mid-Atlantic accent. My father worked at the United Nations, and my mom gave up her job as a secretary to raise the family, and she later became a medical receptionist. She should have been a doctor, but that was not an opportunity open to her. I tried to convince her later in life, but she didn't have the confidence to do that. And she passed away 10 years ago. I'll try not to get emotional about it. Anyway, so because my dad was at the UN, I went to the United Nations school, and I had two absolutely fabulous teachers. Brian Kahn, who was my biology teacher, also anthropology and theater workshop, and Dr. Thomas Sell, who was a marvelous chemistry teacher, and they set me on the path to being a scientist. And while I was at school, I think I was about 16, I read this book, The Double Helix. This is not my copy. It looks pretty similar, but I think mine cost 75 cents and this one cost 95 cents. And I have to say I have to thank James Watson, because James Watson introduced me to Rosalind Franklin. And here's the picture of Rosalind Franklin in the book. And just from reading the book, I was fascinated by this woman who was solving structures and was in the lab every day, and nobody had to tell me that she was the one I should concentrate on. I just did automatically. And I have to say the book really gave an idea of what was the excitement of discovery. Since reading that book, I have come across some pictures of Rosalind Franklin I much prefer. I think she was a rather attractive woman. I think James Watson probably found the least attractive photograph of her to put in the book. And I particularly like this one because I do a lot of microscopy, and so I feel a bit of a relationship there. And of course, everyone knows this photograph. So from school, having been inspired to be a scientist, I went back to England, where my family came from, and I studied biochemistry at Oxford. And from there, I went on to do a PhD at the MRC lab of molecular biology, which we heard about earlier, and I was in Kim Naismith's lab. And I studied, I characterized the first transcriptional silencers, so I was working on transcription in yeast. And towards the end of my PhD, I applied to do a postdoc, and I applied for a Helen Hay Whitney Fellowship And I just want to point out, I was interviewed by Macklin McCarty, which was a huge honor. I was so excited, and he was such a lovely man. It's my favorite interview ever. And I knew about Macklin McCarty because of his work on DNA, best known for his part in the monumental discovery that DNA rather than protein constituted the chemical nature of a gene. And I have to admit, I pulled that right off Wikipedia, but it says it rather well. And this was his famous paper. And he's since passed away, but he was, just the short time I met him, such a wonderful person. So I got the fellowship, and that enabled me to go on to do a postdoc at Harvard with Mark Potashny, where I lasted for a couple of years before moving on to another lab at Harvard Medical School. And I moved to the lab of Norbert Perelman, where I had a marvelous time and developed a system for targeted gene expression, which is used quite extensively in the Drosophila field. So here's a lifeline. So this takes us to just about here. After my postdoc, my partner and I were trying to find jobs where we could both be happy. And there was a point where we thought, well, we could go our separate ways. You know, we seemed to have offers in different places. And we sat down and had to think about it. And I thought to myself, well, I love my science, but I don't want to be miserable doing it. And we made the decision that it wouldn't be worth doing our work unless we were together and happy. So thankfully, we both got jobs in Cambridge in England. And I became a Wellcome Senior Fellow at what's now called the Gurdon Institute. And Jim moved to the, firstly, to the MRC Lab of Molecular Biology and then to the Department of Plant Sciences. And then we moved along happily until just about here, which is when my daughter was born. And life changed quite dramatically. This is my description here. Jim and I didn't think about having kids until I had got my fellowship renewed. I was getting on a bit in age, and I have to say that my mom was diagnosed with cancer, and I think it focused my mind that if I was going to have children, I wanted her to be able to meet my child or children. As it turned out, we were only able to have one, but my mom did meet her for the first 10 months of her life. My family was in the States at that point. Jim's family are in Australia, and so we didn't have the two women under the same roof, but we managed. And I said, it's not been easy juggling two careers and family life, but with a supportive lab and very little sleep, it's been possible, just about. Here's Jim, my other half, who is absolutely wonderful, and there's our daughter from a few years ago, and this is the tea room of his lab. Hannah has spent an inordinate amount of time in various labs, as I'm sure anyone with children will recognize. There was another addition to the family a couple of years ago. This is my dad, who unfortunately was also diagnosed with cancer, and we insisted he move in with us 2 years ago, and he's been living with us in England since then. So we have a three-generation household. So since Hannah was born, science didn't stop. I went on from being a senior fellow to become the Herchel Smith Professor of Molecular Biology, and I've been very, very honored to receive various awards or being elected to various fellowships. So it is possible to keep doing your science. And one thing I wanted to highlight is that in 2006, I was awarded the Royal Society Rosalind Franklin Award, which again was a terrific honor, and particularly lovely for me because I had been so inspired by Rosalind Franklin. And I just wanted to say a couple of things that I did with that award. The award is in part to help promote women in science. And so for my part, I decided to do two things. Number one, I had organized a seminar series inviting women from around the world who worked on neuroscience, and this was primarily for the undergraduates and graduate students and postdocs. So we had a wonderful lineup. Erin Schumann, Magdalena Goetz, Sally Temple, and Christine Holt gave talks. But I also thought it was important to try to target some younger children. And with the help of Nikki Buckley, who's our outreach coordinator at Cambridge, I organized a series of lectures and hands-on demonstration for children aged 9 to 11. And over the course of 3 days, we had 180 pupils, students, in to hear some lectures from my colleagues. We also took them into the lab. They could make their own DNA. They could look down microscopes. And at the end of the day, they would, we took them to the computer lab. They could make their own website to say what they had done during the day. And in particular, we had targeted schools for less privileged children. And it was wonderful. When I walked them into the lab, there was this, just, the sort of sound you'd hear when people go into a beautiful cathedral, but here were children walking into the lab, and that was the highlight of my year. So I just wanted to show you some of their websites. So here's welcome to my website. Explore my pages, and you'll find out loads of stuff about DNA genes and lots more. Here's I Made DNA. We put their DNA in little bottles they could wear around their necks on the way home. This is what DNA looks like close up. Here's a budding geneticist. You're missing the top. Men have the genes X and Y, and women have X and X. So that's why you can't tell if your baby will be a boy or a girl. I want a boy, I want a girl. I particularly like this one. I hope they don't test me. I'm no good at tests. This mouse is going to be tested for all its types of DNA. DNAs are like building blocks of your body. Of course, we showed them some Drosophila as well. Here's a Drosophila. Here are pictures of cheek cells. And I also like this one because when I first heard we were coming to Cambridge University Department of Zoology, I thought it would be really boring, but I'm having a really good time making my own website, and I have learned about cells and lots of other stuff too. And then some of the students were encouraged to send little thank you notes, which I really appreciated, and this one struck a chord. Dear Andrea Brand, thank you for letting us go to the Science Day. We had fun and would like to go again, and we all had fun. Can we go again? From Daniel. So let me see if I can do this now. So I was trying to think of some of my early influences, even very earlier influences, and some of you may remember that there was a film that was showed once a year, and only once a year, back then, and we used to watch it every year, and it was this one.
Brain? That's a very mediocre commodity. Every pusillanimous creature that crawls on the earth or slinks through slimy seas has a brain.
I hope you could hear that. So I sort of think that might have gotten into my brain, and that's maybe why I decided to study the nervous system, but I love The Wizard of Oz. So I'd like to just tell you a little bit now about what we work on, and it is indeed the nervous system. It's the most complex organ in the body, made up of billions of nerve cells. They form patterns of connections, neural networks, supported by non-neuronal cells. And there are maybe several different types of neurons, each with a distinct identity and morphology. So it's a fabulous system to study. But we work on the very, very earliest aspects of neural development. So for example, how do we start off with a group of neural stem cells that are multipotent and are going to give rise to the entire nervous system and end up with a fully functional, mature nervous system? And in particular, as I mentioned, there's so many different types of neurons, and you can see just from their shape that they probably have different functions. So how do we go from stem cells to all this wonderful, diverse array of neuronal types? And so what we're studying in my lab is why stem cells can remain multipotent, how they're able to self-renew, make another copy of themselves at each division, and yet produce daughters, which can give rise to the specialized cells of the nervous system, the neurons and the glia. Now, I don't work on human beings. We work on the humble fruit fly, Drosophila. And the reason for that is it allows us to do a lot of the experiments that we'd like to do. There are 100,000 neurons rather than 100 billion. But perhaps most importantly, if you look at the known human disease genes, 75% of them have counterparts in flies. And the more we study Drosophila, I'm amazed by how many more similarities we see. And there's a drawing by one of my colleagues, Andreas Prokop, which highlights those similarities. And that's here. So here we have a human being, and here we have a Drosophila larva, and you can see how very similar they are. You see they both have brains, and they have motor neurons. And I've said that this is not to scale, so you don't have to worry about meeting a huge larva walking down the street. Okay. And just to show you the actual size of an embryo, this is a British penny, and here's the embryo, so they're quite tiny. Show you a little bit closer. And so we use high-powered microscopes to do our work. But if you open up one of these embryos late in development, this is what you see, a very, very beautiful nervous system. And unfortunately, the brain's been cut off, but you can see these axons, the neurons sending their axons up and down these longitudinals, and motor neurons heading out to innervate these muscles in red. And you can see the connections they make, the synapses on the muscles. So it's a very, very beautiful, aesthetically beautiful, pleasing system, as well as simple and easy to study. And we're interested in three aspects of stem cell behavior: how the stem cells self-renew, We also know that at certain points the stem cells go to sleep. They become quiescent, and we'd like to know what regulates that and how to wake them up when we need to get stem cells producing more neurons again. And we can study all of this in the fly. So here's self-renewal, producing daughters that make neurons. Here they go to sleep at the end of embryogenesis, and then they get woken up and they can reproduce to make more stem cells, but they can also produce neurons. So one of the questions we wanted to address, as I mentioned, is how do stem cells self-renew but also produce neurons? So here's the mother and here's the daughter, and we do say mother and daughter. And in flies, one of the ways that this is done is that the stem cell partitions determinants to its daughter. And so here we've just got chromosomes labeled in red and these determinants in green. So the daughter receives the determinants and takes on the fate of a neuron, whereas the mother stays as the stem cell and keeps dividing. And the determinant that we study is called Prospero. Those of you who might be familiar with The Tempest know that Prospero is the wizard who controls the fates of the other characters, and so that's why this cell fate determinant has been called Prospero. And what we wanted to know was how is it that Prospero stops stem cell division but initiates neuronal development? And I won't go into all the details, but what we were able to show a few years ago is that on its own, it's able to repress the genes that tell a cell to be a stem cell and turn on the genes that are required for cells to become neurons and glia. So it just acts as this beautiful switch between being a stem cell and being a neuron. And what that suggested was that if you removed Prospero, maybe you would never get neurons and glia. And in fact, that's just what we found. So if you remove Prospero, instead of just the mother self-renewing, both the mother and the daughter self-renew, and what happens is that you get tumors. And I'll just show you what that looks like in the fly. So here's a cross-section through a head of a wild-type fly, and here's a cross-section through the head of a fly with tumors. And you can see that in the wild-type animal, here are the eyes, here are the antennae, and there's beautiful patterning of the different cell types in the brain. But if you remove Prospero or various other tumor suppressor genes, you can see this overgrowth, you lose all the pattern, and of course the fly dies. Now, this is of course interesting with respect to human health because there is a hypothesis called the cancer stem cell hypothesis that suggests that some tumors may arise from stem cells which have gone awry. And in fact, Drosophila is becoming quite a good model for studying tumorigenesis. So the other question that we've been interested in, as I mentioned earlier, was why do stem cells become dormant and how can we reactivate them? And of course, this has implications for damage to the brain or disease to the brain if we could wake up the stem cells and get them to produce new neurons. So if we look at the Drosophila nerve cord, this is now the ventral nerve cord, the equivalent of your spinal cord. What we know is that these cells have to grow. The first thing they do once they wake up is they have to grow, and then they can start producing more cells. And so they start off quite small. They're these greeny blue cells, and they have to grow. This is the same magnification, so they have to grow quite considerably. And we know that you need to feed the animals in order for the cells to grow. So if they're starved, you never wake up those stem cells again. So we were interested again in which genes were required in order to wake up these quiescent stem cells. Which are the genes that need to be turned on, which genes might need to be turned off. And we found that the insulin signaling pathway was absolutely required to wake up these stem cells. I won't go into the details, but just to show that there are insulin-like peptides bind to a receptor and initiate a signal transduction cascade which allows these stem cells to grow and start dividing again. And something very similar has been found in vertebrates, again, where insulin-like peptides are able to stimulate stem cell division. So we were curious as to where these peptides were coming from, these insulin-like peptides, to stimulate the insulin signaling pathway in the stem cells to wake them up. And we found that they were expressed by these star-shaped cells, and these are the glia, the glial cells, and these cells sit right on top of the stem cells, and in response to nutrition, they're able to produce insulin-like peptides to reactivate the neural stem cells. And this is just to show that. So when you feed the animals, a signal goes to the fat body, which is the equivalent of the liver in the fly. That sends out another signal, which is actually as yet unidentified. to the glia so that they produce insulin-like peptides to reactivate these neural stem cells so that they can grow and divide. And so what we're interested in then is how we can understand enough about self-renewal of stem cells so that we can figure out what goes on when brain tumors are formed, and more importantly, what to do to prevent brain tumors being formed, and quiescence and reactivation of stem cells so that we can, in the very long term, perhaps regenerate and repair the nervous system. Now I'm just going to flip back quickly to home life. So regeneration and repair is also important for our own minds, and every now and again I realize we should probably have a holiday, and this year I managed to take my daughter to the Sinai Desert, and I can highly recommend, if you want to get away, there is no way you can answer your mobile, there's no email, there's no internet. Okay? So we had a lovely week camel trekking in the desert in Sinai, wonderful. Here's my daughter and her camel. She had a terrific time, and it was also important for her. She grew up a lot, and I think this was the high point. Here she is on top of Mount Sinai, and she's grinning because she beat Mum up the mountain. Okay, and so she grew up hugely on this trip, and we had a fantastic time. I should say this was only possible because my wonderful partner stayed at home to look after my dad. So quality time with my daughter. My dad and my partner had hopefully a good time at home together. I don't know what the symbol is for two men under one roof. There must be one. And I don't know whether it means war as well, but anyway. And so finally, I just wanted to thank all the members of my lab. I'm sorry that everything's been cut off, but I hope you got the message. So these are the members of my present lab, all the wonderful funding agencies who have allowed us to do our research. Here's my lab family, and they really are a family. It's always sad when somebody moves on, maybe happy for them, sad for me. And also I'd just like to thank all my former PhD students and postdocs who've gone on to bigger and better things, and just finally end with Mum and daughter having a good time together. Thanks very much. Are there any questions?
That's fine.
I thought it was fabulous what you did with your prize.
Oh, thank you.
And I have sort of wondered and speculated. I know that women do interesting things with their prizes. I don't think I know of men who do things other than buy cars with their prizes. Maybe, I hope I'm totally wrong, and I would like to hear some rebuttal to that.
Barack Obama gave his Nobel Prize money to the Posse Foundation, which builds links between high schools and colleges for minorities.
He's not a regular.
So do you think that stimulating stem cells to reactivate in vivo is more likely to be the way the field goes rather than isolating stem cells, growing them in culture, and putting them back in?
I absolutely believe that. And actually, I have to say, I find the whole concept of taking stem cells from a dish and shooting them back into somebody's brain rather worrying. Ideally, if there is damage to a particular part of the brain and there are stem cells there that gave rise to those neurons in the first place, those are the cells I'd want to give rise to those same neurons. Whether there's enough information for those neurons to then find the appropriate connections, we don't know. But I think that would ultimately be the best way of trying to repair the brain.
Does your daughter want to be a scientist?
She's only 11, so she does enjoy science. In particular, chemistry, if there's explosions. Apparently, physics is slightly less interesting, and unfortunately, her biology teacher doesn't seem to be very inspiring this year. But she also likes music, and she does wonderful drawings, which didn't come from me because I can't draw at all.
I have one that wants to be a scientist when he grows up, too, and it's the explosions that are definitely a thing. I was struck when you said you did it by without much family support, because that's how my husband and I have done it, and we similarly have done it with very little sleep. And I wonder what you think the long-term effects of that are, because I'm beginning to wonder.
Yes, I'm scared to think what the long-term effects are, but I have to say this morning I was thinking another thing that's absolutely essential is that stuff you put under your eyes to cover the black rings. And I realize I've been doing that since I was in high school, so that's my solution. Okay.